In contrast to the positive social behaviors that are associated with the initial formation of a pair bond maintenance is associated with aggressive social encounters towards novel conspecifics. While sexually naive prairie voles are generally affiliative towards novel individuals, following pair bond formation they become selectively aggressive towards these animals and actively reject them, and this behavior acts to guard the mate and the territory. In the laboratory, we could study this behavior with the use of a resident intruder test. To do these studies, we pair animals for 2 weeks and allow them to mate. At the end of the 2-week period, we employ a resident intruder test by placing a novel conspecific directly into their home territory. If the animal has formed a pair bond, they will aggressively reject this individual, which will be indicated by high levels of aggression. I also wanted to look at the role of the endogenous opioid system in mediating pair bond maintenance. So with our other previous studies, we showed that the mu-opioid system, which is associated with reward and positive hedonics, was important for initial pair bond formation. So for pair bond maintenance, we focus on the Kappa opioid system. And we chose this system because activation of these receptors is associated with stress and aversion. And we thought that activation of these receptors may be key to the processing of novel social stimuli as aversive and the aggressive rejection of these individuals. So to look at the role of Kappa opioid receptors in pair bond maintenance, we selectively blocked Kappa opioid receptors in regions of the brain that we identified to contain high levels of Kappa opioid receptors and that are also associated with pair bonding. So these regions were the nucleus accumbens as well as the ventral pallidum. So prior to tests of selective aggression, we blocked Kappa opioid receptors within the nucleus accumbens shell, nucleus accumbens core, or ventral pallidum. And when we did this study, we found that blockade of Kappa opioid receptors, specifically within the nucleus accumbens shell, but not the other regions, it decreased selective aggressions. This region is important because it's important for processing unconditioned salient stimuli. So what I think is going on is that Kappa opioid receptors within this region are important for processing novel conspecifics as aversive and mediating aversive motivation towards these animals.